Hello everyone, thank you for the opportunity to speak and present us. And we're very glad and looking forward to see you for a third time. And again, each year amazed by the progress that's made. So thank you. So we'll start with a small video.
Selbstständiger wird, insbesondere, dass hier ein bisschen ausdrücken kann, was möchte und was nicht möchte.
I really hope to, at some point, get to a place where Cassidy can, I can just know that she feels good in her own skin.
Naszą największą nadzieją jest postęp w badaniach, który pozwoli lepiej zrozumieć tę chorobę i doprowadzić do skutecznych metod leczenia.
My hopes for Jacques is that he lives a life that is happy, full, and that he's understood.
Dziękujemy z naszej strony za nieustające wsparcie.
Your work means the world to my family and to all of our families.
Gros merci pour tout, puis au plaisir de vous rencontrer.
Thank you for bringing hope to our children's future. Your research gives them a chance at a more independent and easier lives. We are deeply grateful for your commitment, your presence at conferences, and your dedication of time and resources to Argonaut syndromes.
Thank you. And many of the families you had the opportunity to meet are here today and looking forward to see you and answer your questions. And we also brought some little pins, so we invite you later to come and pick up a pin and show that you're part of the team and working with us towards treatment for our children. So the symptoms of Argonaut syndromes, it's a neurodevelopmental disease, so it's very broad. So many children have brain anomalies on MRI scans. Some children have vision problems. Very common is intellectual delay, so learning and understanding information, speech. We also have many children with weak muscle tone, so mobility issues, walking. Some children have something in the heart. Feeding, we also see multiple children, so there's a broad variety of symptoms and also a lot of variability between different patients, a lot of different mutations, so we also invite you to learn more about that at a poster session where you can meet more children and about how they how they experience life, and where their mutations are. Since at the moment we have a count of about 180 patients across the world, across AGO1 and AGO2. We now have more than 70 patients we know of based in the US, also 5 in Canada, now also a fairly big group in Germany and Poland, but we also have families from Australia and Asia, so very really spread across the world. Some of you have seen me before, or Aldona last year, so in 2020, Professor Lessel and Professor Kreinkamp and Professor Meister, they published the paper on the Lessel-Kreinkamp syndrome with the mutation in AGO2 gene. My daughter was diagnosed about 2 months before the paper was officially published. And we then funded the AGO2 Association in 2021 in Switzerland, and we were lucky to get these 3 established scientists on our advisory board and start to collaborate. Also, Professor Piton was at the time, then they were publishing the paper on AGO1 related syndrome. And in 2022, we teamed up and met as well in Regensburg, where there was the first conference where we brought the patients and the scientists together. Then in 2023, we reorganized, so we sort of formally enlarged our umbrella beyond the AGO2 gene to include AGO1. We also had a group form in Catalonia from Anna, that some of you met last year. She had her 3rd child who was born healthy earlier this year, so she's doing well. For those of you who remember her, was she last year pregnant. Aldona, who founded Ago Alliance Poland. Very lucky last year. Victor, congratulations again on your Nobel Prize, and thank you for everything you have done since to raise awareness for the Argonaut syndromes. And glad to be here again today for the 3rd conference and meet you again. What we do is we work with the patients, so to create a support network, peer-to-peer, learn from other families. Many families feel alone and isolated. As you have seen, we're spread across the world, so this is very important. This conference is one of our main events, bringing us together. We are always interested to work with you and collaborate, network. We can help you if you want help to write a support letter or for your grants. Please approach us if you feel that a patient voice would help you in your applications. We've worked with Professor Lessel on the natural history study to collect patient data. And yes, we are doing what we can to help support efforts and also towards model organisms and research. And I will hand over to Aldona, who will share a little bit more about the research.
Thank you. Can you hear me? This is working? Yeah? Okay. So when I joined the association with my son's diagnosis of AGO1 syndrome, having scientific background, I just decided that I want to get more involved and try to push the translational work, because you're all doing amazing work understanding the mechanisms of the diseases, creating model systems, but I asked the question, how can we actually bring the treatments quickly to patients? So the strategy was to scale down, figure out what to focus on to have some starting point. After discussions with Professor Piton and other experts, we found out that AGO1 phenylalanine 180 deletion is the most prevalent. It's also the one my son has, so this was perfect for me to have as the starting point. And then I asked the question, to figure out what model organisms scientists are already using to see if we can use some of the existing strains for the translational work. So I will show that to you in a second. The first step was to consult the strategy, because you have many roadmaps to develop the cure, but the question was, what is the right one for us and how we can actually do that? And then once we've managed to narrow it down, talking with multiple translational researchers as well and foundations, we went on to try building the collaborations, networking, the right partners, and trying to apply for grants together. So when you look at the cure map, we have the first stage that is continuously ongoing with understanding the mechanism of the disease. So this is what you all are doing, and we are very, very grateful for. Then for cohort data, Professor Devereux-Lessel has started the natural history study, which is great help for us, and it's still ongoing. And then there are 4 pathways to go with. You can create like small molecules, you can try reusing the existing drugs on the market, and test them for your condition. You can generate synthetic genes like ASOs, or you can like go with the gene therapy. So these were the paths we went on exploring. And this is the inventory of the strains I've made. So we have the model organisms with 2 most prevalent mutation, phenylalanine 180 deletion and glycine 199 to serine across many of the systems, like worms, fruit flies. We have like fibroblasts and iPS cells. And we put together the application to try getting the mice, because this is usually what's requested by regulatory bodies, although it seems that this year's like it's also changing. And then we also, when we started the consulting and finding the partners, we decided that it would be useful to have the fibroblast biobank. So this is the project that we want to drive as the association for the next year. So now I will show you how we established the partnership and hopefully maybe find some areas for you to join our odyssey to develop the treatment. So there was the paper by Victor and Anna with neurodevelopmental delay worms generated. So since the strains were constructed, I went on to search for the partner who does high throughput screen for the drug library. So once I found it, we applied together for a grant to get the library, and then like this is what we're starting. And once we identify hits, we will go on to test them on like cellular and animal models with help of Amélie Piton or other partners if you're interested. And since these are approved drugs that have the potential to improve patient condition, we can just go directly into administering them. So this should be a relatively straightforward strategy for us. Then the second approach we're going to be exploring, and this is our action plan, is to use the existing knowledge of the structures of AGO1 protein and the mutant, and try to find the binding partner, like design, like using the AlphaFold or some other programs, the small molecule, and then like do the testing and test it and go into clinical trials. So this is like our second stream. And then the third one would be the allospecific knockdowns, which I named like synthetic genes earlier. We're at the stage of like strategy and discussions, but basically we will try to do it like 2 ways, with working on ctRNAs and siRNAs, and just see like where it leads us, because there's no guarantee that any of these strategies will bring treatments straight away, like additional steps might be needed. So using this opportunity, I'm going to show you what are the ways that you can actually support our translational research on AGO1 syndrome. So the first bit, maybe the highest priority for us, is the experimental. We still need some things that needs to be done, like expression threshold ranges, development of robust functional cell-based assays, characterization of mouse models, and development of like AGO1 knocking mice. Then like identification of disease biomarkers and purification of like N and PAS domains of AGO1. So this is our wish list. If you have the capacity or know someone who could do that, please let me know, and I will reach out to the appropriate partners for all sections. And of course, like, we fully appreciate all your support. We would really love if you could spread awareness about our conditions, like share stories at your institutes or at the conferences. Like, we're more than happy to be the speakers also at your institutes for any sort of sessions. For conferences, thank you for your presence, and we would greatly appreciate if you could fill in the questionnaire that we're going to use the information from to try to increase our presence in rare disease community as well. And of course, if you are aware of any potential research partners that could help us with any of these experiments, or you have additional ideas what could be added, reach out as well, and we're always happy to work together on grant applications, and also if you know about any local grant applications for patient organizations, that would be much appreciated, as everything we do is pro bono. All right, so with that, just a quick summary of how you can support us during this meeting. So please come and talk to us. See our posters and videos. This is QR code where you can actually access them online as well. Please fill in the questionnaire. The QR codes are also in the main hall. And join us at the patient panel tomorrow at 1:30. Thank you. Thank you.